Welcome back to part 3 of amino acid metabolism. We will leave transamination and look at the other track of the overview figure on the left. So we'll go this way now. We'll look briefly at the production of ammonium, its transportation in the bloodstream, and its entry into the biosynthesis of nitrogen-containing compounds. Glutamate and glutamine are critical entry points for nitrogen in the metabolism. So recall, glutamate tends to donate its amine group to other amino acids, and its alpha keto acids enter the citric acid circle. At the other end, glutamine generally is a source of ammonia and can enter the urea cycle. So we'll look at this left-hand path in what follows with a focus on regulation of the nitrogen metabolism. So the production and transport of ammonium and its entry into the biosynthesis of nitrogen-containing compounds is a key problem for our organism. Nitrogen is hard to fixate from nature, and on top of that, ammonia is toxic to animals. All the same, most cells produce ammonia to some extent. Alanine is important for transporting it in the form of its amine group in blood to the liver. The liver is schematically represented here. In muscles and other tissues that use amino acids for fuel, their amino groups are collected in the form of glutamate and is further converted to glutamine. So the reaction scheme to the left shows how this is done. Two enzymes are important in this process: glutamine synthase and glutaminase. The latter operates in the liver or mitochondria to release ammonia as ammonium ions. That happens here. These will later be converted to urea in the urea cycle, which will be covered separately in a different installment of this lecture. Now the figure to the right here is a schematic representation of a vertebrate liver. The reaction pathways shown focus on amino acid metabolism. We have already gone through some of these processes in varying detail, so I have blotted them out, leaving only those relevant for glutamine synthase, glutaminase, and the production of ammonia. Glutamine synthase will be the focus of the next slides. It is a very important enzyme with respect to regulating nitrogen metabolism. Glutamine synthase is a primary regulatory point in the nitrogen metabolism. In all organisms, glutamate synthase is either 2x5 or 2x6 identical subunits organized as two rings on top of each other. In mammals, and in the picture shown here, there are 2x6 subunits. The bottom ring is almost hidden, but you can see a little bit of it here. In Trogdor, however, the subunit count is unknown, but almost certainly glorious. Now, glutamine synthase converts glutamate to glutamine. As the reaction scheme presented to the right shows, the conversion is a two-step process. Each subunit has a pocket that accepts glutamate and ATP in the first step. Then, for the second step, gamma-glutamyl phosphate produced in the first step and ammonium binds in the same pocket. Given the importance of nitrogen uptake and turnover, it is no surprise that glutamine synthase is strictly and complexly regulated. This regulation is achieved using covalent modification, gene regulation, inhibition, and feedback inhibition. Glutamine synthase regulation will be the subject of the next few slides. This slide will deal with the inhibition and feedback inhibition of glutamine synthase. Inspect the reaction scheme to the right. After glutamine is synthesized, it serves as feedstock for many other small molecules, including adenosine monophosphate, tryptophan, carboxyl phosphatase, and other product compounds. These end up in the vicinity of glutamine synthase, where they can bind in binding pockets that I have marked completely arbitrarily. This is not a reference to their actual position. using the same crossed out circles that you also find in the reaction scheme. In this way, I emphasize the structural relationship between products and inhibition. In other words, glutamine synthase can sense the level of products it has helped produced and ease of production when levels of these are high. So, in other words, the products feed back into the workings of the enzyme by binding onto the multimeric enzyme. The binding transmits structural change that affects the active sites in an allosteric mechanism. That means there is a transmission of information as the compounds bind through the multimer to the active sites, and this inhibits to some degree the protein. Each inhibitor will only cause partial inhibition, but their effects are cooperative. That is, all of them working together results in more inhibition than the sum of individual effects. Now, in addition to this feedback inhibition, there is also inhibition by glycine and alanine, indicated here in bold. These amino acids are not direct products of glutamine synthase. They serve more generally as a sensor of the state of amino acid metabolism in the cell. Inhibition by glutamine-based products, as well as glycine and alanine, constitutes the first level of regulation, as discussed in the previous slide. The second level of regulation involves covalent modification of the glutamine synthase near its active site. This is an example of post-translational protein modification, where adenylyl is attached to the target enzyme at the tyrosine residue. You can see the modification to the right in this slide inside the blue frame. Both adenylation and the reverse process, deadenylation, is handled by adenylyl transferase. This enzyme, in turn, is regulated by binding of P2 protein. The P2 protein is, in turn, regulated by uridylyl modification. This is done by, and you guessed it, uridylyl transferase, marked UT in the schematic. Uridylyl transferase is inhibited by glutamine and inorganic diphosphate, PPI. Conversely, it is stimulated by the binding of alpha ketoglutarate and ATP. This binding is to the P2 protein, not directly to uridylyl transferase. Now let's try to sum up this. The bottom half of the schematic represents activation of glutamine synthase, while the top represents deactivation. So the two sides of this regulation is connected by uridylyl transferase and the compounds that are regulating it. Now, the uridylated P2 protein also provides a third layer of regulation. It mediates the activation of glutamine synthase gene transcription. Conversely, the deuridylated version decreases this transcription. So what is the net result here? The net result is that glutamine synthase activity is reduced when glutamine levels are high, and increases in activity when alpha ketoglutarate and ATP is high. Now, these multiple levels of regulation allow a sensitive and nuanced response to available resources that fits the cellular need. If your brain is completely pickled now, take a break and return for the next installment.